Here. Okay. Um, Sorry for being late. I literally didn't realize that Marisa is not coming today for class. And I was kind of, yes. Okay. Thanks for reminder. That you see, that's where I need TA. She told me that she won't be here. I just forgot. Let's see. I remember now. Mm-hmm. Yes, yes, yes. That's why I remember when I stopped. I stopped because I saw that people are freaking out. It's already too complicated, right? Let's see. Hmm? I mean, this is really not hard. Nothing is really too hard in this one. It's it's easy, at least for me. But I'm guessing that's because this is what I'm doing. Uh, what did I press? God. Somebody remember where I stopped? Maybe this 29. Aha, aha, aha. That's where we stopped. Okay. I'm going to put it right now. Start slide 29. Okay. Thank you for reminder. Oh, is this where I am? Is this where I stop? Yeah. Yes. Okay. That's Thank you for letting me know, but good luck later on. Everyone will be pissed. I'm kidding. That's what I was doing when I was when I was studying. I would be the one who always tells prof. Yeah, this is what you didn't tell us. And I can see how people so go kill yourself, Marina. Oh. Anyway, so we are still on bioenergetics, and the reason why I stopped last time is because I thought it's very complicated, and I wanted to be very detailed about this. So if, I'm hoping I will be able to finish everything today. But if I feel like it's time to stop, I will let you go because it's really complicated. There is a lot of information, and I would rather give you time to digest than to push just to finish lecture. Is that okay? Now, so we talked about respiration versus like anaerobic versus anaerobic respiration. The common problem that people usually have is whenever you say respiration, you associate that with oxygen and something aerobic. It's not. Respiration means having a specific terminal electron acceptor that will accept electrons from electron donor, and it will get reduced, and then you will get energy out of it. So that means that it will be aerobic if your terminal electron acceptor is oxygen. It will be anaerobic if something else is your your terminal electron acceptor. So this is where actually I maybe just go back one slide to start from electron transport chain in aerobic. So we are describing now, we already talked about this when we talked about four ways of generation proton multiforce. So this is electron transport chain that exists in aerobic respiration when terminal electron acceptor is oxygen. So you will have complex one, where electrons will arrive from complex one, and complex one is called NADH dehydrogenase. Whenever you see the name dehydrogenase, that means that something is oxidized, aka releases electrons, and something else must be reduced, which means accepting those electrons. So complex one, which is NADH dehydrogenase, is oxidizing NADH in cytoplasm. And when NADH gets oxidized by this dehydrogenase, you get formed NAD+. Electrons are moving forward to another carrier, and at the same time, hydrogen ions are translocated from cytoplasm to protoplasm. The second in line in aerobic respiration is ubiquinol, or shortly Q. And ubiquinol is linked to the complex 2. Um, so those electrons that were released by NADH dehydrogenase, they will move back to the quinol, and then quinol will become reduced and become quinol, which means two hydrogen, QH2. Um, Additionally, with complex 2, you will have associated succinate dehydrogenases, which will originate from TCA cycle. What is succinate dehydrogenase doing is oxidizing it's actually oxidizing FADH to FAD plus. So if that's oxidation, that means something else must be reduced. And this is a common in this type of reaction. And those electrons from FADH will be delivered to quinol, and then quinol becomes quinol. Complex 2 is not proton pump, which means there is no proton motive force. I mean, there is proton motive force, but this guy, complex 2 does not pump proton. From complex 2, electrons will move to complex 3. Complex 3 is cytochrome. In this case, will be cytochrome B, and it matters which type of cytochrome because other types of respiration have different types of cytochrome. So in cytochrome B, you will have irons. Cytochrome is famous by having an irons. So irons in cytochrome are oscillating between 3 plus and 2 plus. So iron 2 plus in cytochrome will accept electrons from complex 2, and when they accept electrons, they will become iron 2 plus. They will become less positive, more negative. So this all oxidoreduction on cytochrome is performed by cytochrome oxidoreductase. So that's why it's called oxidoreductase. It oxidizes one thing and reduces another thing. So iron is getting reduced and electrons from quinol are being given up, which means oxidation. F complex 3, which again is cytochrome oxidoreductase and cytochrome B in this case, there is a proton pumping involved, which means it contributes to the proton motive force generation. Next in line is, of course, complex 4. Complex 4 is called terminal oxidase. In this case, it's cytochrome O. So what this complex does is holding the oxygen atom tightly between iron and copper. So you still have iron involved, just in addition with the copper. And it holds oxygen until it's completely reduced to water. So that's a final, and this is direct translocation. If you remember, we talked about that when we talked about 4 waves of proton force generation. Some organisms have different cytochromes. It's not in some organisms you will have cytochrome AA3 or cytochrome A3 or cytochrome B. So there are those variations on team depending on type of organism. Again, oxygen gets reduced, picks up 2 hydrogen ions from surrounding media, and you get water. And the complex 4 also pumps protons. Now, reducing the amount of oxygen. What happens when you have a lower oxygen? So everything what I just described you happens when you have normal oxygen level in the environment. But E. coli can survive in microaerophilic conditions. What means microaerophilic? That means a lower level of oxygen present. So when lower oxygen level is present, not all organisms can survive, but E. coli can. But the way how E. coli does it is switching to the different electron transport chain. So there are different cytochromes involved, so we will cover that briefly now. So I'm not going to go through details through this slide. This is literally repetition of everything what we just said. So everything literally gets the same up to the complex 3, which is cytochrome B and cytochrome oxidase. But there are certain differences. So again, because there is lower oxygen level, you will get lower level of proton motive force. So that means that you have to have different level of cytochrome B extent. So again, with low oxygen available, that means it will be slower growth because there is less material to build proton motive force. So what happens in lower level oxygen is that different cytochrome B complex will be a substrate for cytochrome oxidoreductase. And there will still be proton pumping, but it's just different form of cytochrome B. Now, the big difference is the complex form. If you remember, it was cytochrome O in the normal oxygen environment. This time you have cytochrome D terminal oxidase. What that means? What's the difference? The difference is that this cytochrome D has actually higher affinity for oxygen, which means it will scavenge every single molecule present because there is not enough. So it just increases affinity. So basically, the difference between high and low oxygen environment is in cytochromes in complexes 3 and 4. And what happens at the end of complex 4 is that you will not get proton pumping. So again, you will get less energy. There will be slower growth. So in summary, with low oxygen level, you will get complexes 3 and 4 change. Cytochrome B in complex 3 will express different form, but complex 3 will still be proton pump. And in complex 4, you will have different cytochrome. It will be cytochrome D, terminal oxidase, and terminal oxidase in low level oxygen will not pump protons. So that's the difference. Again, just a mini summary, changes in cytochrome pool and changed PMF generation ability because the last complex in low oxygen does not pump. And now things are getting little more complicated. Again, I won't read all of that in details because pretty much everything gets to be not the same, but similar. Now we are talking about anaerobic. So we went from high oxygen to lower oxygen, now to zero oxygen, like anaerobic respiration. So something else is changing. It's changing the type of quinone. Now you will have menaquinone as a part of complex 2, and it won't be terminal oxidase. It will be called terminal reductase. So everything else will be the same. Like same, like complex 1 will again function the same. What will be different is complex 2, which is now menaquinone. If you remember, with normal oxygen and lower level oxygen, you get quinone that gets reduced and becomes quinol. This time you have linked menaquinone with complex 2. So that's the difference. Now again, you will have different type of cytochrome expressed. So it will be cytochrome B, but different form, different than the one in high and low oxygen. And it will be completely different terminal complex 4, because in this case, with anaerobic respiration, it will be called terminal reductase. What this enzyme does is reducing, I'm giving an example of nitrates because that's common and second in line after oxygen as the energy size to create. So nitrate is reduced to get nitrite. But the enzyme that does this reduction of alternative terminal electron acceptor, which is nitrate, is called terminal reductase, and there is no proton pumping. That is why you will have lower proton motive force, which means slower growth in anaerobic E. coli growth versus high oxygen and low oxygen. And some other so this is just a basic electron transport chains, but you will see in microbes various types of electron transport chains depending on whether they respire. And as you probably remember, microbes will find molecules present in the environment and extract electrons from whatever they find and deliver that electron to something else to create proton motive force. So in this case, we are giving an example of formate that can serve as electron donor, and microbes that use formate as electron donor, they can extract electrons for formate because they can run specific electron transport chain. So the enzyme that drives this formate, so-called format respiration, or formate fumarate, I call it formate. The main key enzyme is formate dehydrogenase. So again, analog to NADH dehydrogenase, what formate dehydrogenase does is extracting electrons from what? From formate. Same how NADH dehydrogenase extracts electrons from NADH. So formate dehydrogenase will extract electrons and move those electrons toward now menaquinone pool. So find that parallel, like compare that with the oxygen. Like you had dehydrogenase, just it was extracting electrons from something else, and you have quinone pool. This time you have menaquinone. When quinone gets reduced, it becomes menaquinone. Again, this oxidation leaves protons behind, and that contributes to proton motive force. Um, this time you will have central carbon metabolism associated with it, and it will be very much clear when it comes to TCA cycle. So microbes from TCA cycle can import fumarate. It's one of the key molecules in TCA cycle, and they also have enzyme fumarate reductase. Fumarate reductase in TCA cycle reduces fumarate and creates succinate. So succinate goes further through TCA cycle, as we will see in a few moments. And this is the way how proton motive force is generated with additional contribution from central carbon metabolism. So remember we talked about succinate dehydrogenase contributing to proton motive force in complex 2. So in this case, in this unusual electron transport chain, you have formate, sometimes can be hydrogen, but let's call it let's not complicate it, as an electron donor, where fumarate from TCA cycle will be electron acceptor with specific fumarate reductase. Again, this is unusual electron transport chain, which I just wanted you to have in mind to understand how there are different ways to generate energy. Now, when when you switch E. coli from high to low oxygen, they will change B types of cytokines and terminal oxidases. Menaquinone pool is associated with nitrate respiration. So menaquinone is a landmark for anaerobic respiration, and it's the only quinone in some pathogenic bacteria because they very often live in anaerobic environment. I am just a summary. Now I'm just giving you a summary, nothing that you didn't hear already. Anaerobic respiration means using something other than oxygen as terminal electron acceptor. E. coli is specific because it can use both. It can switch, but there are certain organisms that simply do not have machinery to respire oxygen as a terminal electron acceptor, so they have to respire something else. So E. coli is, as we know, very well characterized, and it will be able to respire oxygen if you give it to them, because oxygen is the best terminal electron acceptor in tower of power. It has the most positive redox potential. But if you take out oxygen from E. coli, it will still survive. Again, this is just a reminder on what is the best electron donor, what is the best electron acceptor. So every organism uses sugar to create NADHs, and NADH is starting ETC. That's why NADH and sugar start at the top of tower of power. So organisms with compounds with more negative redox potential are donors. Those with a more positive redox potential are acceptors. The best one is on the bottom, that would be oxygen. And higher distance between donor and acceptor, higher differences in redox potential means more energy and more proton motive force. Lower distance, lower energy. And again, the availability of these electron donors and acceptors in environment will actually structurize, for example, the complex environments like soil. Like some organisms will produce one compound that will serve as electron donor for another, while third organism will produce something that will be electron acceptor for the first one. And this is what we will talk about in a few minutes. Again, just tower of power. I'm constantly emphasizing this because I do want you to understand this pairing. Oxygen so if we talk about surface, like oxygen will be on the top. So if you think about Vinogradsky column, like what is on the surface? So microbes that respire oxygen will be on the surface because they respire oxygen, so that they will be exposed to oxygen. The next one in line will be nitrate, because that's the next one in redox positivity. So again, just comparison. So sulfate, for example, is more pure sulfate here. Here, sulfate, sulfur. Sulfate is, if you compare sulfate to nitrate, so sulfate is more redox negative compared to nitrate and oxygen, but if organism doesn't have anything else to respire, if only sulfate is there, then it will respire sulfur, but it has to pair it with something on the top, like probably LDH or organic compound, but the distance will be shorter. So you, and again, you can, organisms can also respire metals. Again, if you look metal here, iron 3 plus and manganese in the same. So iron would be a better electron acceptor than SO than sulfate. Like this one is the best electron acceptor. Then nitrate and iron 3 plus. Then will be fumarate. So pretty much whatever, as you're going from the bottom to the top, this will be the best electron acceptor and then it goes. So this is the most positive on the top and more negative. Yeah, so in finally we should summary. So in summary, oxygen respired first because it's the most energetically favorable, then nitrite, then nitrate, then sulfate, and last carbon dioxide. So microbes use electron donors to extract electrons from an APC. And they generate proton multiforce while they're moving electrons from carrier to carrier, and then gradient content or proton multiforce will run ATP synthesis in the membrane, which is called oxidative phosphorylation. There are other types of organisms that do not run EPC. They're called fermenters. We will talk about them, but they still have to generate some ATP somehow, but they do that through something called substrate level phosphorylation, which would be mini oxidoreduction reactions through internal regeneration of reduction equivalent, but we will cover that in next lecture because we will talk about different ways of ATP generation. Any questions while I am moving to another lecture? I think. Okay. No question. That's okay. Okay. So we're still, again, these two lectures are really interconnected. And this is where we stop. This is where we would stop in previous lectures. So substrate level phosphorylation is a mode of ATP generation that is done by microbes that do not run electron transport chain. So fermentation microbes do not run electron transport chain. Fermenters are those organisms that I would like to talk about now, and many of them are very well known. Again, be very careful. Fermenters do not run electron transport chain. They generate ATP by something called substrate level phosphorylation. And that's a set of mini oxidoreductions, which we will talk about right now. So again, I'm pretty sure you already heard a lot about fermenters. So they can be primary or secondary. Those microbes that ferment sugars to make acid or alcohol, they're primary fermenters because acids and alcohols are their primary products. And secondary fermenters are those microbes that use products made by primary fermenters to generate gases that are important to other microbes. So, for example, those would be methanogens or sulfitogens or acetogens. So, again, those most famous ones that we know about are primary fermenters, like, for example, lactic acid bacteria. So, fermentation literally means a mini redox reaction that occurs while fermenting some organic substrate. And these reactions release very tiny amount of energy, which is going to be just enough to activate certain kinase. You will see which kinase. And what kinase does is phosphorylating ADP to ATP. So that's called substrate level phosphorylation. They generate ATP because that ATP is going to be needed to generate cellular biomass. And very often these fermentation products are excreted. They are excreted and then we can collect it from a media. So Fermenters do have ATP synthase, but they run it in reverse to make proton multiforce to energize transport or flagellum. So ATP synthase does exist, but it runs opposite than ATP synthesis in ATC. In shortly, you will have this organic substrate that will be fermented. So ATP, even though they have ATP synthase, they don't use ATP synthase to create ATP for cellular processes. They create ATP for cellular processes during those substrate level phosphorylation, small redox reactions. But redox state between substrate and product is it's very small distance. Like there is not that high distance like it exists in aerobic respiration. So you still have to have oxidant and reductant. So oxidant would be NAD plus, which is reduction equivalent. And that oxidant needs to be regenerated internally. So that's why product is excreted. And I will show you that in a few minutes when we talk about production of ethanol and lactic acid. So small amount of carbon actually gets assimilated into the biomass. Very big amount gets to be a waste. That's why it gets excreted. Now we did make a use of their waste because that's how we make ethanol and lactic acid. So just a summary slide, what are characteristics of fermentation? Mini redox reaction, substrate and product have similar oxidation state. There will be no electron transfer like we see in alpha okay? And there is no oxidative level phosphorylation, there is substrate level phosphorylation, which means there will be no cytochrome symbols. ATP synthase, different than the one present in aerobic organism, does function to create proton motive force, but proton motive force that is used only exclusively for moving molecules across the membrane or for movement of flagellum, not for biosynthetic purposes. But main ATP is generated by kinase activity, which is called substrate level phosphorylation, where ADP is phosphorylated to ATP. Again, there is oxidant involved. It's NADH usually, sometimes it's NAD plus, sometimes NADP plus. But in order to keep cycle going, you have to regenerate that NAD plus, and you have to do it internally. So again, substrate level phosphorylation is your key word here. This is just an example of some fermentations that you probably heard about. One that we all know is when you ferment glucose to ethanol. That's the beer fermentation. Lactate fermentation, which is lactic acid. And there are mixed type fermentation where you produce butyrate, acetate, and then you can see here hydrogen and carbon dioxide. So there are different types: alcoholic, homolactic, heterolactic, or mixed, or butyric acid, and we will cover some. First in line will be homofermentation or homolactic fermentation. So what is happening here? This is where I will explain in detail the differences with So if you think about homofermentation and homolactic acid, so glycolysis will be present, which means sugar oxidation. So you start with the 6-carbon sugar, which is in the beginning, glucose. In order to activate glucose, you have to phosphorylate glucose, and you do that on that. You remember that complex E2, E2A, B, C that is in the line. So you get glucose phosphate, but in order to phosphorylate that glucose, that phosphate group comes from somewhere and it comes from ATP. So that means you are investing one energy molecule to phosphorylate. Now, glucose 6, I'm purposely not giving you full pathway because it's complicated. I'm just stating that. So glucose 6-phosphate can be glucose 6-phosphate gets converted to fructose 6-phosphate. It's also 6-carbon molecule with a different structural form, and then fructose 6-phosphate gets phosphorylated again, which means you have to invest another ATP molecule, and you get fructose 1,6-phosphate. This is 2-phosphate molecule, which is highly energetic, and this is important for your exam. The key enzyme here that you will have that does not exist in the next fermentation is aldolate. What aldolase is doing to fructose 1,6-bisphosphate is splitting it to 2 times 3 carbon molecules. So fructose 1,6-bisphosphate has 6 carbons. When you cut it on half, you get 2 times 3 carbon molecules. One will be glycerol-3 phosphate, the other one will be dihydroxyacetone phosphate. This one is very hard to pronounce, I call it DHAP. Why they matter? Where did you see DHAP? Lipid biosynthesis. When we talk about glycerol biosynthesis, not GLPS, internal. So glycerol-3 phosphate and dihydroxyacetone phosphate are 2 trioses, so 3 carbon sugars. Now, both of them will now go through the same process where this one will be glycerol-3 phosphate will be, and this is important, this will be oxidized. So you can literally think about two lines going from two molecules because everything happens in phase. So glycerol-3 phosphate gets oxidized by an enzyme called GAPPH, and it's not shown here. It's because I don't want to bother you. What happens there is reduction of NAD plus to NADH. So this is that internal reduction of NAD plus NADH, NAD plus reduction. The product of that oxidoreduction, when NAD is reduced and these two guys are separately oxidized, is 1,3-bisphosphoglyceric acid. Important compound. Why? Because once you have this 1,3-bisphosphoglycerate or glyceric acid, in the reaction with kinase, here you get ADP phosphorylation to ATP. That's substance level phosphorylation. And then you have serious reaction where this bisphosphoglycerate is converted to 3-phosphoglycerate, then 2-phosphoglycerate, then phosphoenolpyruvate. Nothing of that I'm showing here, so I reduced it maximally. But bottom line is to reach from phosphoenolpyruvate, you will get this pyruvate, but in order to get that pyruvate, you have to get pyruvate kinase, so this here would be PET or phosphoenolpyruvate. Then you will get stripping of that phosphate group from phosphoenolpyruvate, creation of ATP from ATP by kinase, so kinase, you get pyruvate as a product. And you have 2 ATPs, and you get 2 molecules, right? So you technically have 4 ATPs, but you invested 2 in this process, so your net gain is 2 ATPs. So now this is a pyruvate. What you do with pyruvate? If this is happening in anaerobic conditions, what you would have this pyruvate sent into the converted to acetylcoenzyme A, and that acetylcoenzyme A would go to PCSI. But because you don't have an ATC here, what do you need to keep this cycle operating? Think about from energy perspective. You need to get the way to make ATP. The key to get that is to keep this cycle operating, because you have to regenerate energy force. How would you do it? The only how fermenter can do it is to evolve the enzyme called liver lactate dehydrogenase, or later on we'll see ethanol dehydrogenase. But that enzyme will do. That enzyme can dehydrogenase will oxidize now made NADH to NAD plus and reduce pyruvate to black. Okay? And then this NAD plus can go up and gets reduced again so the cycle can operate. That's called internal regeneration. And this product of lactate is our, the lactate is our product that we use internally, lactic acid. And the same thing happens, it gets excreted. Same happens with when you have ethanol dehydrogenase. So lactate dehydrogenase and ethanol dehydrogenase, or alcohol dehydrogenase, sorry, are not there to make nice stuff for us. They're there to help bacteria to regenerate oxygen internally. Does that make sense? Then glycerol-3 phosphate and dihydroxyacetone phosphate are interconvertible. Very often, when you see DHAP overly expressed in some organism, that usually means that cell is going through some sort of membrane-associated stress because it's switching from glyceraldehyde 3-phosphate to DHAP, because DHAP can be used to make more phospholipids so it can remodel membrane. So if you're doing metabolomic analysis and you see very high level of DHAP, that tells you that your cell must be going through some membrane-associated stress, like acidic, alkaliphilic temperature, etc., etc. So is that clear? And these, so in that, I'm not going to read all that because this is given to you only so you can, everything what I just said is in these slides. So I just thought it would be better if you guys have that in writing. So when you read, you don't have to really catch what I said. You can have this in writing. But this is literally everything that I just explained in that. And this is just a smaller version. We emphasize enzymes that I do want you to know. And I did not mention some of them, so good that I emphasize. So you remember when I said fructose 6-phosphate becomes fructose 1,6-phosphate? So the enzyme that does that is quite important. Why? Because that's regulating that. So if you don't get fructose phosphorylated to become fructose 1,6-phosphate, there will be nothing that aldolase can break. There will be no glycolysis. There will be no. So PFLK is going to be the key enzyme here, one of them, because it's a regulator. GALPH is the one that produces NADH, and those two kinases are important. So I just emphasized what I think you should know, but the most important will be PFK, lactate dehydrogenase, and aldolase that splits fructose 1,6-bisphosphate. Why aldolase matters? Aldolase matters because when you have heterofermentation, which is mixed products. So if you remember now, we had only lactate produced and lactate dehydrogenase. What is the true heterofermentation? They have different products in addition to lactate. So that means they don't have they have similar enzymes, but it's a slightly more common. So as you can see, you will still start with the glucose, and you will still have glucose phosphate, but you don't have here. You don't have because there's no aldolase. You will have no fructose 6-phosphate. You will not. You won't have that PSK. No, you will have glucose kinase that will give you glucose 6-phosphate, and then because there is no aldolase, you have to overcome somehow that issue. So you will. You will have this enzyme that will convert glucose 6-phosphate to 6-phosphogluconic acid, and you are producing here NADH. But what you have to do here, you have to go from 6 to 5 carbon molecules. So it's slightly less energetic in favor. That game is fine. So now you have ribulose 6-phosphate and carbon dioxide, and ribulose 5-phosphate is actually a pentose phosphate, like 5 carbons. Get the isomerized, it doesn't really matter. What matters is that from 6 molecules, you go to 5 carbons, and then what you have, you now from 5 carbon, you somehow, the whole point is to get glyceraldehyde 3-phosphate or dehydrogenase 3-phosphate, because those trioses can go down to produce ATP. How will you get that? If you have 5 carbon molecules. So CO2 went up. Now you have glyceraldehyde 3-phosphate and acetyl phosphate. Acetyl phosphate seems a little tricky. What will you do with that? Glyceraldehyde 3-phosphate is easy. That one will go through the standard pathway. Lactate will be produced. There will be lactate dehydrogenase, and NADH will get recovered. Over. You have 2 ATPs produced from that. Now, what you do with acetyl phosphate? Again, cell is fermenting, so it has to find a way to make a use of. So then you will go around and you will develop additional dehydrogenase, which will now oxidize NADH, again, you are regenerating NAD+, reduce acetyl phosphate, and then you get acetaldehyde. And then there is a second dehydrogenase, and I'm purposely saying only dehydrogenase because I don't want to bother with the compound names. But the point is that this dehydrogenase is now again oxidizing NADH and reducing acetaldehyde to ethanol. So you have ethanol production. So that's how alcohol gets made. So all purpose is to get NAD plus regenerated. And what will you do? You have glycerol 3 phosphate, easy peasy. You will get a lot from that. What can you do with acetyl phosphate? You have to convert it to alcohol and then you excrete that alcohol and then we drink. If we don't have classes. Yeah. Yeah, I was thinking about that. Yeah, but I was told that that's real. Yeah. Anyway, so again, I'm not reading all that. This is just writing for your record so you can read at home, like just to like so you don't have to try to catch what I said or to try to understand my accent. Everything is written. You can just read it, and if something is unclear, so I said all of this. You just have that in writing. When I was a student, my former boss never wrote on slides. He was just talking, and he's Russian, so I barely understood him. That's how I learned that I will actually write it down. So whenever you are at home, you can read it and enjoy, because I really enjoy reading this. This is something that I love. I could do this for the rest of my life, and that's exactly what I will do. Uh, anyway, so lactic acid and ethanol are important for biotech purposes, and if somebody is in micro 315, you probably heard about, you will hear a lot about these things. Um, Fermentation and heterofermentation. So, lactic acid bacteria. Who took 322 last year? How do we call lactic acid bacteria? To which family they belong? I see Mike already thinking I'm teasing you. I don't really expect you to know that. I'm just teasing you. They're called firmicutes. I'm just teasing you. Why would you remember that? If I have to remember, you don't. Anyway, but lactic acid bacteria are really, they eat lactose fat and protein milk and they produce lactic acid. Uh, they usually give they usually produce some sort of fats like amino acids, and because it's a they give low pH, they produce organic acid as well. That gives like a very acidic taste, and it's called protected fermentation because of low pH. Like nothing else can grow in that low pH. So uh, common fermentative organisms are usually doing fermentation of that mild cheese, like a mild flavor and acidity of the cheddar cheese. But if you see these like a sweet cheese, like with the bubbles, that's usually heterofermentation because of CO2. They produce CO2 as a gas, and their gas gives those bubbles. And now make it more complicated. If you were not sick enough of homo and heterofermentation, meet cost review. That one is doing butyric acid fermentation, which is even more complicated. But again, I will tell you exactly what is the key point to know. So, how these things function. Of course, there is no cytotoxins involved, and it's a mixed type of fermentation where you have products acetate and butyrate. And those are early fermentation products, acetate and butyrate. And when acetate and butyrate are produced, like acetate, when they are produced, like acetate and butyrate, when they get produced, that lowers pH. When like low pH happens, that will stimulate different set of genes, and now organisms will consume butyrate and acetate, and they will make butanol and acetone. Again, depending on what you want, you can produce only acetate or butyrate, and then you stop fermentation, you get the products out. You don't let them go up to the butanol and acetone. But you could, because if you have acetate and you have butyrate produced, which is up there, that will lower pH, and that lower pH will stimulate butanol production, and for acetate, you can get to acetate, and you can even go to isopropanol. So again, all what you need to know is basic stuff in this one. I never ask this in detail because it's overly complicated for you to remember. In other words, I will never ask you to know all these compounds if you're scared of that. Another type of fermentation is quite interesting, and I love this one, and I always ask this one because it's important for our mouth, you know, because I'm personally suffering from that a lot. I really want to know. So it's called Strickland fermentation, which is fermentation that involves amino acids. So all what you need to have one more redox negative electron donor amino acid, and the other one that is electron acceptor, which will be more redox positive. And I give you examples of some amino acids that can cause fermentation. I don't want you to memorize which one is donor, which one is acceptor. I do want you to know that Strickland fermentation involves two amino acids. One needs to be electron donor and it's redox negative. The one that is electron acceptor will be redox positive. That's all what I want to know, in addition to substrates that are required for this fermentation to happen. So again, you need electron donor and electron acceptor. So two amino acids, doesn't matter which one it is. Like if I ask a question, I will tell you which one is donor, which one is acceptor. Or I will tell you highly reduced amino acid, and then you have to know that if something is highly reduced, that is electron donor. And you need to have ADP and PI. Why ADP and PI? Because those are substrates to generate ATP. Remember, the purpose of fermentation is to generate ATP via substrate level phosphorylation, not to make beer for us. As a product of those short reactions, you will get ammonium, carbon dioxide, and acetate, and the most important ATP, because ATP is that's why they are running this type of fermentation. What is the significance of Strickland reaction? It prevents dental caries. So problems that you can have in mouth. So where is the problem? So in our saliva, we have proline. Proline is amino acid. And Peptostreptococcus is commonly plaque bacteria. And what it's doing is completing fermentation of glucose to produce free amino acid. So in our mouth, what's happening is this Strickland reaction that promotes the reaction between free proline and amino acids made by plaque bacteria in our mouth. The product of that fermentation is the valeric acid, which can be degraded to ammonia. And ammonia has a specific role because it removes protons and reduces acidity, and acidity is cause of dental problems. So this is a useful reaction in our organism. Strickland reaction. Any questions about fermentation before I move to central carbon metabolism? So central carbon metabolism, for people who are doing metabolism for grad students specifically for doing metabolism, this is quite important for them. And it's easy because this central carbon metabolism is for almost all microbes, like pretty much everyone. Not everyone, but it's very similar to everyone. So basically, the point is to process carbon skeleton in cytoplasm, which is the basis for catabolism and then like metabolism. What is the purpose of catabolism? To generate energy, literally. And you generate energy through either oxidative phosphorylation or substrate level phosphorylation. And then that energy is later used for metabolic purposes. And also, you have another type of metabolism, which is called anabolism, which means generation of metabolites for biosynthetic purposes. Again, the way how you will recognize catabolic versus anabolic reaction is in catabolic, this is not written on slide, this is from live class. And there is a reason why I mentioned it. Catabolic reactions will always have production of ATP, while anabolic reaction will have consumption. Prokaryotes actually have very diverse metabolism. So, and it's very diverse comparing to eukaryotes. Like we have only aerobic metabolism. Prokaryotes, and again, if somebody took biology to talk about metabolic diversity, all of that. So literally, they can use anything what is out there to extract electrons and generate another compound. So I was using glucose as example here because that's universal. And again, it's not all microbes can grow on glucose. There are those oligotrophs that can. But glucose will be something that I will point out here because, again, it's the most common, it's the most universal. There are different variants of glucose degradation, which is glycolysis, and I will cover all three of them. But again, this is just when we focus on E. coli mostly, but there are organisms that cannot metabolize glucose as a sole carbon source. So we will focus here on glucose degradation simply because it's very universal and it's the most common and it's very well known. It's very well characterized. So there are glycolytic pathway glucose degradation can be called EMP or Emden-Meyerhof. I call it EMP. But also there is a version of glycolysis that is called EDD or Ethner-Dodorov pathway because I don't know how to pronounce that very well. I call it shortly. So it's enough to know EMP, EDD. Pentose phosphate pathway is another version of glycolysis, and all of them are favorized depending on nutrients that are given to the microbes. So microbe can use each of these central carbon pathways to reconstruct carbon skeleton in cytoplasm, generate energy, and generate biosynthetic compounds that will be used in anabolism, and energy will be used in catabolism. EMP pathway. Again, pretty much something that you already saw when we talked about glycolysis in fermentation. What is the difference? So again, I already mentioned the phosphofructose. So when cell is deciding, when cell is favorizing EMP pathway, you will know that based on the expression of certain enzymes. One of the key enzymes will be PFK, rate limiting step, and it's irreversible. That means once when PFK happens, you can go back. Key molecule, fructose 1,6 bisphosphate. And then you have this downstream that we already talked. That will be EMP pathway. Pyruvate then gets converted to acetyl coenzyme A, and acetyl coenzyme A goes to TCA site. And this is, again, just a writing for you. I'm not going to read it. The most important step, phosphofructokinase, irreversible step, and this is just in details. So we kind of talk about this already when we talk about fermentation. Just in fermentation, there is this stage, pyruvate. Pyruvate has various states. State of the pyruvate in fermentation is to go to lactate, to be reduced to lactate or ethanol, and then we get the specific production. When you have anaerobic respiration, pyruvate gets converted to acetyl coenzyme A and goes to TCA cycle. We will cover that too. What are important points? Again, this is literally the same writing that I had in the previous slides, just I thought it will be useful for you to have that in case you want to read in multiple levels, because I needed it. So again, this is the different kinds here. We talked about technical pyruvate. If you pyruvate will go to acetyl coenzyme via hydrogenase, and acetyl coenzyme is going to be metabolized to TCA cycle. We will cover TCA cycle in next slide. In TCA cycle, as you will see, there will be creation of NADH uses through series of reactions. And there is also NADH from glycolysis created, and all of that will now go to TCA cycle, ETC cycle, sorry, which is going to happen in the membrane, which is oxidative phosphorylation. But if there is fermentation, there will be no TCA cycle. Now, how to the EMP pathway is EDD. Sorry, this should be EDD, not EDP. Ethner-Dodorov pathway. Where is the key point here? So the key point in Ethner-Dodorov pathway. So if you remember, this has similarities to heterofermentation. So you still have 6-phosphate and 6-phosphate acid. But there is no alcohol. There is 6-phosphate acid that will be converted with dehydrates to this, let's call it 2-KDPG because it's really complicated. This one, 2-ketone-3-deoxy-6-phosphate acid or KDPG. This is still a metabolite that indicates existence of EDD cycling bacteria. Why is this molecule important? So this KDPG is analog to fructose 1,6-phosphate because you will have aldolates, again, just different type of aldolates, not the one that breaks fructose 1,6-phosphate. This one will break KDPG. And when you break these two, you have literal dextrose-3-phosphate and fiber. You see, this is already useful enough and you can run metabolism. Literal dextrose-3-phosphate go to 5-beta-ethanol or can go to DCA cycle. This fiber can also go to ethanol DCA cycle depending on the type of the organism. So usually in organism has choice to do fermentation or to do aerobic distillation. But the point is that you came from 6 carbons to 2 times 3 carbons. When you had fructose 1,6-bisphosphate, you had literal dextrose-3-phosphate dehydroxyacetone. This time you had literal dextrose-3-phosphate and pyruvic acid or bio. So, but the way what indicates presence of EDD pathway is KDPG, because that molecule really tells you that it's that this bacteria is running EDD pathway. And again, just writing that everything what I just said in details, so you can figure out better what are, so you can understand better what's going to happen next. This glyceraldehyde 3-phosphate can also be used in pentose phosphate pathway, which we will mention in next slide. So we usually, this EDD is called modified glycolysis. And organisms prefer this pathway depending on the nutrients, at least those organisms that we work with. Some organisms have ability to run either or. Ximomonas, for example, uses only EDD, and it's big competitor for yeast fermentation because it's doing fermentation of starch into the biofuels and bioethanol. It's better than yeast because it produces ethanol faster and it has better tolerance to ethanol. So Ximomonas with EDD is the competitor for yeast in ethanol fermentation, but it's very hard to maintain it comparing to yeast. Another one is pentose phosphate pathway, and the name tells you it's all about pentoses and phosphate. And what you need to have in this pathway is you have to get, you will recognize it by specific different, so you will have sugars that have 4, 5, or even 7 carbon molecules. So the way how this functions is through sets of isomerases that are generating different versions of carbon skeletons through isomerization. And it usually, this process usually functions simultaneously with glycolysis because glyceraldehyde 3-phosphate can also be sent to pentose phosphate pathway because many of these pentose sugars, like ribulose or 5-phosphate, is used for nucleic acid synthesis. So mostly carbon is processed through glycolysis, but some portion, like 20%, has to go through pentose phosphate pathway, mostly for biosynthetic purposes. And also pentose phosphate pathway is the only one that generates NADPH in oxidation reduction processes. Again, I'm not talking about photosynthesis. Photosynthesis is a different method. Pentose phosphate pathway has 3 main parts: oxidative, non-oxidative, and isomerization. Oxidative part of pentose phosphate cycle is including conversion of glucose to ribulose or 5-phosphate, and that usually involves decarboxylation because glucose is 6 carbon, ribulose is 5 carbon, and then you have 1 phosphate added. Then you have to isomerize this ribulose or 5-phosphate, like just changing the structural formula. And the non-oxidative portion, which is rearrangement of sugars depending on what is needed for biosynthetic drugs. And we will see in the next picture what means rearrangement of sugar. So again, it starts from six molecule, then six, then one carbon goes out, as you can see. So glucose is six, then six is here, then you have pentose phosphate, then you have isomerase, pentose phosphate isomerase, which will just change from riboso to riboso 5-phosphate. So it's just different structural formula. Then you have epimerase, and then you have cellulose. Cellulose is also 5-phosphate. So pretty much you have isomerization of these forms. In one moment, you will have erythroso 4-phosphate. And why would you need all of these molecules? So glyceraldehyde 3-phosphate, as you can see, the one with 3 carbons, that one can be used later on in biosynthetic process. It can go through phosphoenolpyruvate, give pyruvate in the end, pyruvate converted to acetyl coenzyme A, acetyl coenzyme A goes to this cycle. Fructose 6-phosphate can also go to glycolysis, be phosphorylated, and then you get fructose 1,16-phosphate, and you again become allolase and cut glyceraldehyde 3-phosphate and PHAP. You make those two from 1,16-phosphate. Then you have this set of capsule 7-phosphate and erythrose 4-phosphate. So this one has 4 carbons. You might wonder why do we need it. Erythrose 4-phosphate is usually used for synthesis of aromatic amino acids. One of the key landmarks that will tell you when you analyze metabolomics, it will tell you that your organism is running pentose phosphate cycle is going to be 7 capsule of 7-phosphate. That one is also used for amino acids, but that's a landmark of pentose phosphate pathway. And again, aromatic amino acids from erythrose, pentose sugars are using for nucleic acids. NADPH is used for lipid synthesis, glyceraldehyde 3-phosphate and fructose 6-phosphate used for glycolysis and PCA. And this is just the same thing like a writing of pentose phosphate pathway. Same, pretty much the same like I did for the previous one. Key metabolites in pentose phosphate pathway: pseudoheptulose, erythrose, ribulose. The reason why I put them in slide is because I want you to know them. So this is like a summary. So pseudoheptulose is the less important that it's used in lipopolysaccharide biosynthesis. More important is that pseudoheptulose 7-phosphate is the landmark of pentose phosphate pathway. Erythrose 4-phosphate is used for aromatic amino acid synthesis. It doesn't matter which one. I'm not going to tell you which are aromatics. Just know that. But I'm assuming you know that it's cyclopropane, alanine, and tyrosine. Ribulose 5-phosphate used in carbon dioxide fixation if organism is doing it. If not, ribulose 5-phosphate get isomerized to ribose 5-phosphate, which is used for nucleotide biosynthesis, ATGCs. And glyceraldehyde 3-phosphate, which is abbreviation G3P, and fructose 6-phosphate, which is FP6, is used in glycolysis and PCA sites. So again, it's just now. I am thinking it's 12:10. I will finish this last slide, and then I will let you go before you all speak out, because this is really extensive. But the reason why I want you to see these metabolomic studies is because sometimes in metabolomic studies, not sometimes, always in metabolomic studies, we can determine whether cell is using EDD, EMP, or pentose phosphate pathway. And the way how we do that by labeling carbon, and then we track it through HPLC or GC or through some specific device. And then you can see, you can determine, or the easier way is just to analyze metabolomics, which is my specialty. So you label glucose with C40 and you track. If you see that you're detecting in HPLC or mass spec pseudoheptulose, that means that carbon flux goes through pentose phosphate pathway. So in E. coli, most of the carbon through glycolytic pathway, very small amount through pentose phosphate pathway, but in xenomonas you will see pseudoheptulose coming out labeled a lot. To determine which metabolic pathway is used, you have to track the key metabolite of each mode of each pathway. So, for example, if you want to see whether it's EMP, EDD, or pentose phosphate pathway, you label carbon with C14 or C13, and then you are observing in HPLC or MS which molecule is showing up. If you see accumulation of cytokaptose, okay, it's mostly pentose phosphate pathway. If you see fructose 1,6-bisphosphate or you see a lot of pyruvate, that would be EMP. If you see KDPG, that's EDD pathway. But you track it and you can see that you can use this tracking to discriminate between EMP and EDD pathway. And this is where I'm thinking to stop. So, and it really differs. Like it's easier for us for doing labeling with, so we work with molecule that is called methane and it has only one carbon. And that's the only one that we can label. We can't do anything else. When you have molecule like glucose, then you have 6 carbons that you can label. So it's going to really depend where you want to label it. You want to label carbon position 1, you want to label 2, 3, 4, 5, blah, 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 whichever you want. But depending on where you label carbon, you will see different compounds. That's where the trick is. And in my defense, this question was earlier on exams when Dr. Stein was teaching, but I really didn't think students can answer because it's really hard. It took me a while to figure out. So if you label carbon on position 1 of glucose, so glucose has every atom is labeled like you have position 1, 2, 3, 4, 5, 6. So you label carbon on position 1. And then, glucose get processed through all glycolysis, and then either EMP or EDD. And depending on how the process goes, you see different accounts. You see label on different positions. So if it's labeled at position 1, and you see labeled acetyl coenzyme A accumulating, that means your organism is running in. But if you label on position 1 and you start detecting carbon dioxide labeled, then that means it's EDD. Because carbon dioxide, so that carbon that you label radioactively or fluorescent, in EDD pathway gets removed in decarboxylation. And then you will see it in CO2. And you only detect labeled compound. You don't detect that. But then you can do another thing. You can label carbon on position 3 of glucose, and then situation is different. In that case, you see accumulation of acetyl coenzyme A, but you label it position 3. That means organism uses EDD pathway. And if you see labeling in carbon dioxide, that means it uses EMP. So it really, the key point is that it really depends where will you put that radioactive or fluorescent label. And citric acid cycle is something where I will stop now because there is so much to know there and it will be overly complicated. So we are going to talk about TCA cycle and we will start potentially unit 7, depending on how it goes. We will see. I don't want to overwhelm you. This is already too much. Okay. So let's stop here and we will move on next class.